traditional sequencing and NGS, then the steps involved in NGS, then pyrosequencing. These are the types of NGS, that's next generation sequencing, then Illumina sequencing, solid sequencing, then comparisons of all these NGS types, then application and limitation of these techniques, then how we can overcome through TGS, that is third generation sequencing, and its application as well as the limitations, then future prospectus of this sequencing techniques. Coming to the introductory topic, that is what is DNA sequencing. First of all, if we split this term DNA, that is deoxyribonucleotide, that means it contains sugar, then base and phosphate. The bases can be adenine, cytosine, guanine, and thymine. And the technique that is to use to determine the nucleotide sequence of deoxyribose nucleic acid, that is DNA, is called as sequencing. And it is the fundamental level of knowledge of a gene. Then brief history of DNA sequencing. It is as the 1952, the DNA structure was photographed by DNA diffraction method by Rosalind Franklin in 1952. Then the 1953, the famous Jameson-Watson-Crick, that is Watson-Crick double helical structure, what we know today, it was introduced in 1953. Then DNA sequencing plan was started in 1965 by Sanger and Robert Hawley. Then around 1977, they have refined that Sanger's technology of DNA sequencing. That been commercialized in 1987 by Applied Biosystems, and the second generation sequencing was started by around 1996. Then it has been commercialized in 2005 by Roche 454, and then Solexa has been started in 2006. Then 2011 it has been commercialized. Then the next part is third generation sequencing, that is long read sequences were unveiled out, and it has been commercialized in between 2011 to 15. So this is the history of DNA sequencing, and coming to the methods involved in this, the well-known methods like Sanger's sequencing, Maxim and Gilbert sequencing, these are traditional methods involved in this. And coming to the NGS, second generation sequencing, this is Illumina sequencing, pyrosequencing, then solid sequencing that we are going to cover in detail today. Then apart from this, the third generation sequencing is SMRT, that is called as single molecule real-time sequencing, and ONT, that is Oxford Nanopore technology. In addition to this, what are quoted here, further innovations and further changes have been taken place, and these are only brief classification, I can say. There is further research is going on, and further expansion of these sequencing methods are going on. Then traditional sequencing, the basic idea behind that is in Sanger's methods, that is chain termination. This is it is anyways it is the synthesis, that is sequencing by synthesis, it is called as, where the chain termination, that means chain terminators are used that are having no 3-OH group, that is they are lacking of 3-hydroxyl group, so that there is no continuation of polymerization by DNA polymerase could take place. Then Maxim and Gilbert method, this is involving DNA sequencing by chemical modification, and this method is here a brief picture of this Maxim and Gilbert sequencing. Here the 5' is radio labeled, and single-stranded DNA will be cut into different pieces, fragments by G site, G plus A, T plus and C, and cytosine sites by using different chemicals. So these chemicals will able to cleave this DNA, and those cleaved DNA fragments will allow to run on a electrophoresis in gel, and then the sequencing will be deduced. DNA sequencing will be deduced by this reading that sequence. Coming to the Sanger sequencing, this is still popular because the method used here is still extrapolated for other NGS or TGS also. Here, the thing is, I already told, that is dideoxynucleotide sequence is used, that is having no 3-hydroxyl group There is no hydroxyl group at 3' end, so there is no polymerization reaction can continue. It is wherever, for example, if adenosine triphosphate is added in the sequencing, that it can't extend further, so it has to halt that. So like this, DDCTP, DDGTP, and DDTTP are used, and the halting of the polymerization technique will be taking place. And then it is allowed to run on a gel electrophoresis. Then whatever sequence is reduced here, this is a sequence of complementary strand, because this sequencing through, that is synthesis method, and complementary strand is read by 5' to 3' from downward to upward. So this is all about the traditional sequencing methods, and still that is followed in especially Sanger sequencing. So then coming to the next, that is what is NGS. NGS is nothing but next generation sequencing. It is the new technology for DNA and RNA sequencing, and it is helping for identifying the variants in DNAs. It is a combination of different sequencing matrices and bioinformatics, and it is having a massive parallel sequencing of various lengths of DNA and RNA. Coming to the difference between traditional versus NGS, traditionally single DNA fragment at a time can be run, or it can be sequenced, whereas in NGS, billions of fragments can be allowed to sequence. NGS is helping more number of samples can be screened at a time, whereas the traditional methods, it can't be possible more number. It is only few number of samples can be allowed. And it is highly sophisticated technique, NGS is, that's why it is highly sensitive, and it can detect multiple variants. And but the traditional method is not suitable for identifying the variants. And it is low sensitive, and the limit of detection is around 15 to 20%, whereas NGS is highly sensitive. And one more thing is, it is cost effective if the sequences are more than 20, whereas it is not that much cost effective for minimum sequences, the traditional sequencing method. It's not that much cost effective for smaller sequence. Coming to the steps involved in NGS, the first step is nucleic acid extraction, then library preparation, then sequencing as such actually, then the interpretation technique, the data analysis and interpretation by bioinformatic tools. This is exactly, the NGS is not so easy as we mentioned, or as we talk, because it's every molecular technique has to be screened up to this sequencing, and further the data analysis and bioinformatic tools application and interpretation of the result is it needs a lot of patience and understanding of the background of NGS. Then coming to the first step, that is DNA fragmentation. DNA is broken into small pieces of around 700 to 800 base pair length. The breaking methods are two, that is especially mechanically and enzymatic methods. Mechanical methods, two methods are involved: ultrasonication and nebulization. Ultrasonication here is high frequency sound waves are used to cut the DNA fragments, DNA into smaller pieces. Whereas the nebulization is the high flow air is allowed to break the DNA into small pieces, while it is allowed to pass into a nebulizer by high pressurized air. At that time, what happens? It is a nebulizer is containing a small hole on which the DNA is allowed to pass. At that time, the breaking will be taken place. Coming to the next technique, the next method is enzymatic method. That is, restriction enzymes are allowed to cut the DNA into small fragments. Then, of course, the transposon nicking enzymes are used here. They produce overhang of 5' overhang or 3' overhang. Then randomness of fragments and overlapping is, this is a very much important step in DNA fragmentation through enzymatic method. And it is required also for library preparation. The next step is library preparation. Library preparation is to identify the DNA sample by tag. Here, the sequencing adapters are added to each DNA sample. Then corresponding sequencing primers will bind to this, and because the primers and adapters are parallel to each other, so they are able to bind each other, and the sequencing will be done. The ligated adapters to the DNA will be selected either by electrophoresis or they can be taken by the magnetic beads, and then the sequencing will be done on a planar surface. Coming to the emulsion PCR, this is actually the principle behind the pyrosequencing method. This is NGS type of NGS. In the next slide, I'll go first and then I'll come back to explain this, because otherwise it will be, there will be some confusion will be there. So this is pyrosequencing principle. Here the dNTP, that is a nucleotide sequence, this will be in the presence of DNA polymerase, it will be broken down into polyphosphate. Then APS. Here the APS and luciferin are substrates, and the single strand DNA is hybridized with a primer. Enzymes are DNA polymerase, ATP sulfurase, apyrase, and luciferase. The ATP sulfurase is helping to produce ATP by polyphosphate, and the luciferin will be produced. In the presence of luciferase enzyme, this will be cleaved into AMP and polyphosphate. In addition to this, the oxyluciferin light is emitted. So here the polyphosphate sequence is cleaved off and the light is produced. This will be detected by the cameras, and it will be able to identify each nucleotide upon the departure of the polyphosphate. So this is the idea behind the pyrosequencing principle, and emulsion PCR method is involved in amplification. Emulsion is nothing but a mixture of oil and water, where the capture beads are used, which are having oligonucleotides, and it is complementary to the adapter sequence. Here, this is a picture of an emulsion, oil and water emulsion. This is water and droplet technique. So here, oil and water emulsion will be agitated so that one droplet on which one bead is there, and in one bead, the primer is attached, and complementary to that primer, adapter and DNA fragment will be bound. And then in this each droplet, in each bead, I can say there is enzymes, like DNA polymerase, then nucleotide sequences, then not nucleotide sequences, various nucleotides, magnesium ions, in order to take place the PCR reaction or amplification reaction. So this act as a bioreactor. So each bead is having multiple DNA fragments through amplification. And for pyrosequencing, this all amplified fragments will be, that is double-stranded DNA will be separated into single strand, and this each bead is having many single-stranded fragments, and then they will be allowed to pyrosequencing. And this pyrosequencing will be taking place in a picotiter well, that is picotiter plate this is, and this is called as each of these wells of this picotiter plate. And it is allowed to, that is suspend one droplet or one bead into one well. And so that is taken care here. And then the pyrosequencing, as I told the principle, it will be carried out. Then pyrogram is produced based on the light produced, and it is detecting the different sequences, different nucleotides. That is nothing but a sequencing. So this is the method of pyrosequencing. In total, I can say the picotiter plate and picotiter well, the DNA that is capturing bead having one single DNA fragment that is allowed for pyrosequencing is there in this. So the principle behind this is luciferase technique, and that is used to detect the color. So next is Illumina platform, that is sequencing method. Illumina, that is the bridge PCR is used here, DNA denaturation and then adapter ligation, then amplification in the flow cell. Here the reversible terminators are used, and they are able to halt. They are not permanently halting the polymerization reaction. They will temporarily, the polymerase is blocked, and then subsequently it can be extended. Then laser is used to detect the fluorescence, and based on the color produced or fluorescence light produced, the DNA nucleotide sequencing will be done. So DNA in the earlier, the procedure is DNA denaturation will be taking place, and around 700 base pair fragments, base pair lengthy fragments will be broken. and those will be ligated to the adapter. Then in the flow cell, rather than bead in the case of pyrosequencing, the flow cell is used where the primers are attached. So complementary to the primer, the DNA fragments which are having adapter that is complementary to the primers are allowed to run on a flow cell. Then there is extension of this amplification takes place. Whenever there is a two strands will be produced, the forward strand, there is two strand that is double-stranded DNA. In this original DNA template is washed up, on only a single strand is allowed to run on the flow cell. Then what happens? Whatever adapter is there, that is finding the suitable complementary match, that is whatever single fragments are there here, they will find the suitable complementary to that adapter or primer to this flow cell, and they will start bending, and then the polymerization will be taking place here. So like this, they will bend and produce that. This is called as bridge PCR. Though this amplification is taking place like this, and afterwards this DNA denaturation will be taking place. All the reverse strands will be cleaved off, and only the forward strands will be remained. And by this time, what happens? Each round of amplification, lasers are passed over the flow cell and to activate the fluorescent label on the nucleotide base. The fluorescence is detected by the camera and recorded by a computer, and each of the base gives different colors of fluorescence. Thereby, the base calling or color to base identification can be done. So this is the idea behind Illumina sequencing. Next is solid sequencing, that is sequencing of oligonucleotides by ligation and detection. So this is a bit different technique because here ligation methods are used here. As generally what happens, the target sequencing will be there, and to which the adapter is ligated. And then complementary to the adapter, the primer is attached. And this much till this, it is common to earlier methods. But here the new thing is here probe is attached to this. The probe is attached in such a way that the probe is not like other probes. Here it is having some specialities in this. The specialities of this probe is the first two sequences of this probe is having complementary to the target sequences. Whereas the next three are degenerated bases, then last three are universal bases. So I repeat, the first two are complementary to the target sequences, rest three are degenerate bases, and rest three are innocent bases, or they can called as universal bases. So this probe of having 8 mer sequences, that is 8 nucleotide bases are there. So this is allowed to run attached to these target sequences. And one more thing, interesting thing is here the probe is having a 5' end, which is having a fluorophore. And this is having the color, which is as per the dye base present over here. That means, for example, if AT is there in this complementary initial two nucleotides, complementary to the target sequences, then that color will be different. And if TT is there, the color may be different. CT is there means the fluorophore color will be different. So this will be the technique to attach. And then cleavage agent is, once it is bound, then ligase enzyme is helping to join the sequences. And then last three pair base pair, however they are innocent bases, the matching is not that much complementary. Those will be cleaved off. And this repeated ligation cycles will be taking place. And then the next round of primers are attached to this, and this takes on and on. But the point is, a point involved here is second round of primer, one nucleotide lesser to the earlier primer. That means if primer, first round primer, if it is having n sequences, this is having n-1. That means one less nucleotide to the earlier primer it's having. So this is repeating cycle will be continued like this. And it is interesting point here is it is a dye-based color identification. I already told that, for example, if blue fluorescence is out, that means the first base is T and the second base is C. So If G, I mean green color is produced, means first base is G and it is second base is C. So this is the dye-based color identification based on which the matching will be done, cycle 1, cycle 2, because the primers are shorter to the earlier primer. So repeated analysis will be helping to sequence the DNA. And of course, the emulsion PCR is used, that is as earlier discussed. So the comparison of NGS types, that is Roche 454, that is pyrosequencing platform. This is running time is for single day. Then Illumina sequencing, it is taking around 10 days. There is a reversible termination is principle behind in it. Then solid sequencing, here the sequencing by ligation, running time is around 8 days. Then rest, whatever there, they are modification of earlier methods used. In addition to this, live ion torrent is there. This is hydrogen ion semiconductor is used here, and based on hydrogen ion movement here, the detection will be done. Anyways, these are many modifications have been taken place further, and but basic ideas are 3 platforms are mainly used. Coming to the applications of NGS, these all principles and stories of NGS types methods are over now. Now where it can be used, because ultimately the application part is important. Whatever biochemical tool, new techniques that might be there, but where it is applied and how the cost effective, that is the important thing in all new technologies. So application in antiviral drug resistance. So the thing is that how the antiviral drug resistance or antimicrobial resistance genes present in bacteria or any other genomic sequences, this can be identified by sequencing method by NGS. So like quality control of live attenuated vaccine in diagnostic virology, here any contamination from a original DNA sequences of vaccine viral sequences that can be done through NGS. That means sequencing is done by NGS and then the quality control can be checked. Then if there is any mutation is there or is any SNPs, then that will be bringing out the disease. Those can be analyzed by this. Then targeted panel sequencing here. Suppose some cancer analysis or cancer detection technique, no need to analyze whole genomic assays. That means whole genomic sequence is not required there. If particular part of particular organ, for example, if lung is affected and lung cancer has to be detected, at that time only particular gene has to be sequenced, whether there are any deviation or any abnormalities there. There is no need to sequence entire whole genome, assay, no need of that. So only targeted sequencing also can be done by this NGS. Then prenatal testing in order to check the baby's health in advanced version before the delivery of the baby, that can be checked. Even in veterinary science also it can be done. And sequencing, if there is any mutation, then we can derive any conclusion. Then application in veterinary sciences, NGS was used to analyze domestication pattern of the horses, because when the horses moved from the wild to domesticated, the pattern of lifestyle, then of course there is a modification in the gene structure and genomic evolutionary changes have been taken place. And those can be checked. The mutation studies, copy number variation or any SNPs, those can be checked by sequencing by NGS. And sometimes what happens, it is helpful to resequence to check mutation, because already some genomic sequencing was done for a particular one bacteria or for animal or buffalo or anything. That time NGS may not be there. So that might be done by traditional method of sequencing. Then it can be rechecked. Now any mutation study for recheck for mutation by resequencing. So this is even helpful to study or sequencing viral genome from the post-mortem sample, and this can be used to prepare viral vaccine for wild endangered species. So because some endangered species, if they found dead in wild, so reason to be analyzed. If there is any viral attack or there, then from the genomic sequences, viral vaccine can be produced. So that can be done through NGS only, because sequencing and analysis. Then coming to continuation of application in veterinary science, eggshell-related genes can be sequenced by NGS. This has been done in 49th week Rhode Island red hens. The calcification of the shell and its pattern responsible for any mutation, any changes, any transcriptomic, those all part can be done by NGS. Sequencing of wildlife, African white lion, Asian tiger, white Bengal tiger, and snow leopard revealed that they are closely associated genetically. So this will be helpful for understanding the evolutionary changes that has been there in wildlife or across the distant species. So this NGS supports the discovery of new bioactive natural products without culturing the microorganisms. The thing is, suppose one bacteria is producing some bioactive natural product, which is helpful for helpful might be as a medicine or any other product that might be useful in human industry or human medicinal value or pharmaceutical industry. It might be useful. Those, if we want to have, means need to culture the bacteria, then that product has to be isolated, then it has to be tested. No need to do all these things. If NGS is done, then sequencing will be helpful to analyze whether that is able to produce the new bioactive natural product. So there is no need to culture the microorganism as a whole, and NGS is helpful to identify. Then RNA sequencing also can be done for NGS. That means NGS can be applied in RNA sequencing also. It is helpful in RNA splicing studies, structure, and SNPs. The transcripts of somatic cells of milk, mammary tissue are analyzed for lactation, and any differences and changes, those can be checked by this. Then mRNA protein structure for meat quality facilitates for biomarker analysis to predict traits, and if there is some structure is which was good trait character, then that can be helpful for further breeding purpose or further breed generation, etc. All those genetical studies can be done. Then myostatin gene was found downregulated in horses after exercises, and it can be related to the developmental or metabolic process or structural differences. So this is also important one point. However, this is useful in many sequencing methods, and it is applicable in veterinary science in many places. There will be limitation in any technique. If there is advantage, of course, some disadvantages will be there. We can't have anything 100% and perfect. There is nothing perfect in universe. Even in human beings, we can't be perfect 100%. So that's a limitation of NGS. That read length, when complex repetitive regions are there, means it can't read, because the read length is very small. And if there is a GC bias, that means continuous GC sequences are there, means we can't read. Then, of course, this NGS is short read analysis. That means the time is very much required. And specialized bioinformatics tools are required to analyze, and complicated post-processing pipelines and data storage are very difficult. So that's why the limitations are, this is all not, any NGS or genomic sequencing is not that much cost effective. So that is the one big issue in this. Then overcoming to this limitation, that one more technique has been come out, that is third generation sequencing. The third generation sequencing is directly sequencing of single molecules that can be able to read longer reads. That means more than 10 kilobase per can be read. And the platform Pacific Biosciences and Oxford Nanopore Technology, they will produce this. They will able to sequence by third generation sequencing, that is, they have been commercialized. Here, the flow cell with zero waveguide is used in single molecular real-time sequencing, that is SMRT we say. And in the nanopore sequencing technology, nanopore is inserted into the electric resistant membrane. The idea behind it, the Pacific Biosciences, here the flow cell is used, zero mode waveguide we call, and the DNA polymer is embedded in this membrane-like structure. And the DNA fragment is allowed to run on this, and the fluorescence, whatever produced, is allowed. It is analyzed, and it is called as base calling. And the fluorescence, which is producing for a particular nucleotide, one fluorescence, that will be detected by the cameras and recorded through computer. Then coming to the library preparation is like NGS, it is a bit different. Here the hairpin adapters are used. This is to prevent the breakage or any damage. And this is called a smart bell library generation. Then sequencing will be done. The next is Oxford, ONT we call, Oxford Nanopore Technology. Here This is nothing but here the DNA fragment is allowed to move in a electric resistant membrane, and the motor protein is there that is helping to guide the DNA fragment, that is single stranded fragment. And the reader protein, once the DNA fragment or DNA 1,1 base pair, if it is coming inside this resistant membrane, there will be alteration in the electrical resistance. So this alteration in the electrical resistance is different for one nucleotide. For example, if the thymidine, it is different from cytidine or something like that, it will be there. So the electrical changes, alteration in the electrical resistance, that will be denoted by these cameras, and then computer will be recording that. So these are the just same thing whatever I told, that is SMRT, that is single molecular real-time. That is SMRT flow cell is equipped with the zero mode waveguide, and DNA polymerase is fixed here. Laser and camera detects the color and signal. Then circular double-stranded DNA are the adapters. ONT, that is motor protein for guiding, and reader protein is helping to detect the alteration in the electrical current. Coming to the application of TGS, it is used in genomics to detect large structural variants and de novo assembly of the repeated regions to identify tandem repeat number. Coming to the transcriptomics, this is differential studied here. Then annotated splice isoforms can be identified. Then single molecular level characterize RNA isoforms. Epigenomics, that improves methylation status, and it is overcome bisulfite treatment to analyze the epigenomic pattern or methylation pattern. Then metagenomics establish specific microbiome profiles, recognize mixed microbial communities, and perform targeted gene analysis. The continuation of the same, it is helpful to study the transcriptomics, because full-length transcript variants and gene fusions can be studied by TGS, where NGS can be missed. NGS may miss some transcriptomic studies because of splicing, alternative splicing methods. Those smaller read length may miss some it is not able to give a full whole transcript because of a smaller reads, but this can be overcome by TGS. It is a direct sequencing and direct detection of epigenetic modification, and the longer reads represent most part of the chromosome. And here longer insertion, longer deletion can be identified. There is no PCR bias because there is no the PCR technique is involved here. It is direct sequencing methods. There is no PCR bias, and homogeneous genome coverage will be there. Then limitation of TGS, this is error rate is more than 15%, and definitely it is not suitable for SNPs. Small single nucleotide polymorphism can't be analyzed by TGS. This is only NGS is helpful for this. And higher cost, of course, no sequencing, higher version, higher genomic sequencing is always cost. It is costly only. The fresh samples are required because longer reads are prepared. That's why the fresh samples will produce good results. That's why it can't be done by, or the quality is compromised if the frozen sample or preserved samples are used. Coming to the future prospectus or future directions, I already told any sequencing methods is not that much cost effective. So the progress in bioinformatics tools, progress in robotics, progress in nucleic acid preparation, any special bioinformatic tools, they have to be identified in future so that the cost can be reduced. And that it is always a collaborative approach, biochemist, biotechnologist, bioinformatic experts, then statisticians, then biologists, all has to be sit together and jointly they can organize and derive a conclusion here. It can't be done by single personal approach, or multi-omic integration is possible only by all put together. It is a collaborative manner it is possible. So if whenever these bioinformatic tools, specialized techniques are evolved out, and if they are able to reduce the cost, and if these techniques made cost effective, and then if they are able to popularize among the future in the field, then only this will be helpful to the future. Otherwise, it will be a research and explorative studies can be done by this. So exactly to coming to the field, to the veterinary animal part, that has to be made cost effective. So in future, there will always scope in further inventions or further betterment in technologies. So there is definitely 100% chances are there to reduce the cost of all those techniques. So there is a better future in animal husbandry if this is been done. So these are the references involved, and thanks for giving me opportunity to the NADCL organizer and the Atari team Pune. I'm highly thankful to all, and thanks to the participant for their